Okay, so anaerobic respiration, nitrate reduction lab, experiment number 28. So, background real quick. We talked about respiration. So, yeah, a lot of these like physiology experiments about respiration and how the organisms can use energy, because there's so many different ways that you can use energy sources in microbes, and this is just a taste of them. If you really want to get to know like how many there are, you have to take something like a prokaryotic diversity class or something like that. There's so many different. It's a lot of different ways, and they're all really interesting. It's really cool. But anyway, we talked about fermentation. We talked about oxidative respiration a good bit. Now we're going to talk about going to be talking about anaerobic respiration. So just to make it full circle. So what anaerobic respiration is, it's respiration, but without the final electron acceptor being oxygen. So we talked about the electron transport chain, being the final electron acceptor being oxygen. This, instead of being oxygen, is just something else. So it's other molecules like sulfate, carbonate, nitrate, and there's a whole bunch of different other ones. So there are microbes that take advantage of nitrate reduction. So right here, this is the nitrogen reduction kind of pathway. Like this is like the reduction steps of nitrogen, right? So you start off as nitrate, all right? Then you can use nitrate reductase to go to nitrite. So nitrate reductase to nitrite, and then you can use nitrite reductase to go to I guess that's nitrogen oxide, I think. I'm not too good with my nomenclature in inorganic chemistry. So, but then that nitrogen gas can go to molecular nitrogen, and then that molecular nitrogen can be reduced further to ammonia. And ammonia is kind of smells a little bit. That's the smell of urine. So, yeah. But you don't necessarily have to go through all of these reductions, right? So you can start off reducing just nitrate to nitrite, and that's it. You can just do all that, or you can just reduce nitrate to NO2 or N2O gas, right? Or you can go all the way to ammonia. Like you can start at nitrite and then go to ammonia, or as long as you're going this way down the reduction pathway, you can use any kind of combination you want. So what happens is that these microbes that can do this nitrate reduction, they live in dirt. As you can see, kind of some of these end pathways are gases. So what can happen is it's called denitrification. It's when the gases in the pathway go into the air, and what happens is you remove the nitrogen from the dirt that stuff's growing in. So if you don't have any nitrogen, then you cannot grow in the soil. Crops need nitrogen, whether it be for amino acids, proteins, whatever it may be. Amino acids and protein, nitrogenous bases and proteins, or whatever it may be. Some even phospholipids. But you need it to grow. So if you take away the nitrogen, you can stop the growth in like a particular field from that dirt. And that's why you have to replenish nitrogen. So if you ever heard of a farmer having to rest a field, or you do crop rotations, or there's a ton of different ways you can correct for this phenomenon, denitrification process. But they're talking about, like, the reason why you have to rest a field is to replenish the nitrogen. That's the limiting reactant of growth, if you will. So what we did was we tested for reduction. All right. So what we did was we inoculated nitrate broth, and we had a nitrate broth tube and it had a Durham tube in it. All right. And so it has this nitrate NO3 minus in it. And we inoculated it with microorganisms. All right. And what we did was we kind of tested. So they're going to take that nitrate and reduce it down somehow. And so what we kind of did was we kind of tested backwards. So the first thing that we're going to test for is the reduction all the way to ammonia. And the way that you do that is that ammonia smells like urine. So what you want to do is you want to do like your little whiff of the hand and, kind of whiff it toward your nose and see if it smells like ammonia. Don't panic. It won't smell like ammonia. Like, they're not going to do that to you, right? But it's something that you would test for. It's going to be negative, but you just want to do it for kicks and giggles, all right? So the next thing you want to see is if it reduces it down to a gas, because if you, so you're testing, the first one you're testing all the way here, the next one you're testing all the way to NO2 and NH, or N2. So we don't know which gas it kind of went to, but it can go to either one. They're both going to be gas, and they're both going to float up on that Durham tube. So if you have gas in your Durham tube, you know that they went all the way down to a gas. The next thing you do is you test for reduction to nitrite. So it's nitrate going to nitrite. You're going to test for that. And what you do is you're going to add alpha-naphthalamine acetate and sulfonilic acid. All right, and so these two are reagents that will react with nitrite, and if there is nitrite in there, then you're going to have a red color being produced. All right, if it did not reduce down to nitrite, then you will have, it's just going to stay the color, it's going to stay this like not red color. All right, I forgot what color it was. I haven't done this in a while. But what you do is you want to always check yourself. So you're going to have a control tube, and you're going to want to add zinc to that control tube. All right. So what zinc plus does is it does the reduction of NO3 to NO2. So it does this reduction. What that means is you still have that alpha-naphthalamine acetate and sulfonilic acid in the tube that you added zinc to, so it does that reduction, and so it will then make a red color. If there's no red color made after you add zinc, then you did not have any nitrate to begin with. So it's kind of like a troubleshooting experiment, and or to make sure that you kind of had that stuff in there. So if there's no red color produced after you add zinc, then something is wrong. Okay. So the results are that, like, if you have smell, then it reduced it down to ammonia. If you have gas, it reduced it down to this stage. If you have red, it reduced it down to this stage. And if you had to add zinc to get the red, then it had no reduction on the nitrate. All right. And so the takeaway is that we just talked about anaerobic respiration, how you can use things other than your oxygen to accept the electrons of the final electron acceptor. Once they are accepted from that final, once they accept the electrons from the electron transport chain, they are then reduced, and they are reduced down to some level. What level it is, we do these tests for. All right, and so you can use this, you can test for these reductions by using either the smell or the Durham tubes, or different reagents, whatever it may be. But it just gives further indication of how microbes use energy and their physiology.